morning and welcome back to the channel. It's beautiful to have you back here with us. This morning we are talking about triple sugar ion test. That's the TSI test, triple sugar ion test, which is also one of the tests that is used in biochemistry to differentiate organisms. Now it's used majorly in that same family, Enterobacteriaceae. It's used to differentiate gram-negative organisms within that family, one from the other. So we can use it to differentiate Shigella from Salmonella. So these are important. This is an important test, and this test is quite important because it has three steps, or it is interrogating three different steps. One is interrogating the ability of the bacteria to ferment different types of sugars: glucose, lactose, and sucrose. So it's investigating the ability of the sugar to produce to ferment or make use of any of these three kinds of sugars. Secondly, is looking at when the bacteria is making use of this kind of sugars, does it produce gas in the process? That's the carbohydrate fermentation. Does it come with gas production? And the third thing that is checking is this bacteria, is it able to make use or to produce hydrogen sulfide? Is it able to make use of sodium thiosulfide and use it to produce hydrogen sulfide, H2S? Now, so these three different cardinal points are what our triple sugar ion test, that is what it stands upon, and it's important for us to know. Now, there is a media, the TSI media, that is prepared. The media is made, in the media you have the composition of the media is made of glucose, lactose, sucrose, phenol red, an indicator, peptones, sodium thiosulfate, and ferrous sulfate. These are all what you have, and then agar for solidification. Now, it's important to know that in the TSI agar, when the slant is made, there's an aerobic part of this slant, and there's at the bottom of the preparation of the media, you have an anaerobic part. And so when you prepare the agar, then you have the organism of interest. You stab it into the agar. That's the method of inoculation when we do a TSI test. You stab it, especially into the anaerobic portion. Remember, I told you that there are two portions of the agar. There's the aerobic portion and the anaerobic portion. So you stab your bacteria of interest. The way you inoculate it is to stab it into the anaerobic portion of the agar, of the media, and then you streak onto the aerobic portion of the agar. Then you begin to watch. For different kinds of color changes. And like I said, the first one is sugar fermentation. Is it able to ferment glucose, lactose, and sucrose? You look at that, and if it does that, it will produce an acid. An acid will be produced which will turn the medium yellow. So the medium will turn yellow, and acid will be produced if it is able to ferment any of these glucoses. Secondly, when is it able to utilize peptones? If it's able to utilize peptones, it will produce an alkaline byproduct, so it will give you a red coloration. Thirdly, gas production. When it breaks down these sugars, is it able to produce gas? If it's able to produce gas, you will see the bubbles of the gas. You can hear crackles, you can see the bubbles that will show you that, oh, gas production is taking place here. And then the third one is H2S production. Is it able to make H2S by making use of sodium thiosulfate, which is present in the media? And if it does that, then it will produce a black precipitate. The black precipitate will be produced, which you would see with the eye, which will be an indication to tell you that sodium thiosulfate has been used up. And like I said earlier, there's an indicator substance that is used, which is phenol red. Make use of phenol red here. And so when it is yellow, we know that this is acidic media, or acid has been produced. If it is red or pink, we know that it is alkaline. So these are important when we prepare the media and then we inoculate. Then we incubate at between 35 to 37 degrees for 24 to 48 hours, and then we bring it out and view, and then record our results. Which make when we record our results, the results, if it is yellow, we know that that's acid has been produced, so that's carbohydrate fermentation. If it is red, then it means that alkaline was produced and we had peptone catabolism, meaning that it was unable to break down the carbohydrate. If you see the black precipitate, that tells you that sodium thiosulfate has been used up, and then gas bubbles have been produced. So these are all important. And like I said, you can use it to differentiate your Salmonella, your Shigella, your E. coli, and your Pseudomonas. It's a very important test and it has a lot of advantages. It helps us to differentiate enteric bacteria, helps us to identify different types of bacteria, and it's important. Now, it also has its own limitations. Some organisms are not able to produce very strong acids, so you can be getting false negative results. Meanwhile, those organisms are actually supposed to be positive. So the TSI test is the very important test among our biochemical battery of tests. It's good that we get to know it, get to know how it is used, how the agar is prepared, and the different things that we do to make it to make use of it. Thank you for listening to this. Remember, like, share our content, and we'll continue to grow this community. Have a wonderful day.